Hello students, this is your instructor Abid John, and today's lecture is about amino acid properties and consequences of substitutions. Introduction. Since the earliest protein sequences and structures were determined, it has been clear that the positioning and properties of amino acids are key to understanding many biological processes. For example, the first protein structure, hemoglobin, provided a molecular explanation for the genetic disease sickle cell anemia. A single nucleotide mutation leads to a substitution of glutamate in normal individuals with valine in those who suffer the disease. The substitution leads to a lower solubility of the deoxygenated form of hemoglobin, and it is thought that this causes the molecules to form long fibers within blood cells, which leads to the unusual sickle-shaped cells that give the disease its name. Hemoglobin is just one of many examples where single mutations can have drastic consequences for protein structure, function, and associated phenotype. Protein features relevant to amino acid behavior. It is beyond the scope of this lecture to discuss the basic principles of proteins, since this can be gleaned from any introductory biochemistry textbook. However, a number of general principles of proteins are important to place any mutation in the correct context. One of the features is protein environments. The second is protein structure. The next, protein evolution, and the last one is post-translational modification. Protein environments. A feature of key importance is cellular location. Different parts of cells can have very different chemical environments, with the consequence that many amino acids behave differently. The biggest difference is between soluble proteins and membrane proteins. Whereas soluble proteins tend to be surrounded by water molecules, membrane proteins are surrounded by lipids. Roughly speaking, this means that these two classes behave in inside-out fashion relative to each other. Soluble proteins tend to have hydrophilic residues on their surfaces, whereas membrane proteins tend to have hydrophobic residues on the surface that interact with the membrane. Soluble proteins also come in several flavors. The biggest difference is between those that are extracellular and those that are intracellular. Cells also contain numerous compartments, the organelles, which can also have slightly different environments from each other. Proteins in the nucleus often interact with DNA, meaning that they contain different preferences for amino acids on their surfaces. For example, positive amino acids for interacting with the negatively charged phosphate backbone. It is important to consider the likely cellular location of any protein before considering the consequences of amino acid substitution. The next is protein structure. Proteins themselves also contain different microenvironments. For soluble proteins, the surface lies at the interface with water and thus tends to contain more polar amino acids than one finds in the core of the protein. Proteins also contain regions that are directly involved in protein function, such as actiocytes, in addition to regions that are less critical to the protein function and where mutations are likely to have fewer consequences. It is important to remember that the context of any amino acid can vary greatly depending on its location in the protein structure. The next is protein evolution. Proteins are nearly always members of homologous families. Two processes can give rise to homologous protein families: speciation and duplication. Proteins related by speciation only are referred to as orthologous, and as the name suggests, these proteins have the same function in different species. Proteins related by duplications are referred to as paralogs. To be maintained in a genome over time, paralogous proteins are likely to evolve different function. Differences in function can range from minute differences in substrate recognition to complete differences in cellular location and function. The next is post-translational modification. The gene-specified protein sequence can be shortened by proteolysis or lengthened by addition of amino acids at either terminus. Many diseases arise by abnormalities in post-translational modifications, and these are not necessarily apparent from genetic information alone. Properties of the amino acids. Amino acids can be in more than one group, and that sometimes properties as different as hydrophobic and hydrophilic can be applied to the same amino acid. Hydrophobic amino acids. Amino acids that don't prefer to be in an aqueous environment. The hydrophobic can be divided according to whether they have aliphatic or aromatic side chains. Aliphatic side chains. Strictly speaking, aliphatic means that the side chain contains only hydrogen and carbon atoms. By this strict definition, the amino acids with aliphatic side chain are alanine, isoleucine, leucine, and valine. Aliphatic side chain are very non-reactive and are thus rarely involved directly in protein function, although they can play a role in substrate recognition. Aromatic side chains. A side chain is aromatic when it contains an aromatic ring system. The strict definition has to do with the number of electrons contained within the ring. Generally, aromatic ring systems are planar and electrons are shared over the whole ring structure. The amino acids with aromatic side chain are phenylalanine and tryptophan. Aromatic residues have also been proposed to participate in stacking interactions. Here, numerous aromatic rings are thought to stack on top of each other. Polar amino acids. Polar amino acids prefer to be surrounded by water. Those that are buried within the protein usually participate in hydrogen bonds with other side chains. Aspartate and glutamate are negatively charged, lysine and arginine are positively charged. Small amino acids. The amino acids alanine, cysteine, glycine, specifically proline, serine, and threonine are often grouped together for the simple reason that they all are small in size. In some protein structural context, substitution of a small side chain for a large one can be disastrous. This is the bibliography for the above text. Stay calm. Allah Hafiz.